Hello, everyone. This is an exam 2 review for genetics. In this review, we're going to go over X-linked recessive diseases, heritability, epigenetics, genetic linkage. Um, yeah, and in addition to those topics, you'll also need to know about the required reading. So the first thing to talk about are the differences in X and Y chromosomes, specifically how they relate to X-linked recessive diseases. Remember, males are XY, so the Y chromosome contains the SRY gene, which encodes info to turn the embryo into a male, meaning that a loss of function mutation at this gene could cause abnormal sex characteristics. And there's a lot of different variations here in the phenotype. I encourage you to look into that. So the two X-linked recessive diseases you have to know are hemophilia A and red-green color blindness. So this is associated with loss of function. To identify it on a pedigree, look for males having a higher chance of being affected than females. And two phenotypically normal parents can have an affected male child. Keep in mind that an affected X allele is never passed from father to son, since the father has to pass a Y chromosome to the son. Therefore, if you have an affected son, the affected X chromosome came from the mother. And daughters of affected fathers are at least carriers, if not affected. And that's explained here. So I've crossed an affected father with a carrier mother. And you can see the different probabilities here. So the probability of having a daughter who is a carrier is 50%, and the probability of having a daughter with a disease is 50%. So we have no phenotypically normal daughters here who are not carriers. And then the probability of having a phenotypically normal son is 50%, and the probability of having a son who is affected is also 50%. So just always try to draw Punnett squares for those in case you get confused about the probabilities. Moving on from X-linked recessive, just some basic definitions. So remember that the phenotype of an organism can be influenced by several different things. It's based on the organism's genotype, its environment, and the epigenetic modifications on that genotype. A quantitative trait is a measurable phenotype, something like height, and a quantitative trait locus is a region on a chromosome corresponding to the genetics of the quantitative trait. Variation can be from several different quantitative trait loci, causing a spectrum of phenotypes. Like this is related to epistasis. Major affected QTLs are genomic loci, which have a very strong impact on the phenotype, so they are more influential than other QTLs. And some more definitions that you have to know. Multiple alleles. So this is where many variants of a phenotype can occur, which that definition is pretty general. So if you can remember the example here, which is a pretty good example of what multiple alleles means. So the example is cystic fibrosis, which as we've learned is Mendelian, but the locus is very large. So there is kind of a lot of room for mutations to occur. There's many different mutations that can cause cystic fibrosis. So if you remember back to that little like pamphlet thing that we read with the different classes of mutations for cystic fibrosis, that's how we can get so many different classes and still consider it to be cystic fibrosis. Codominance is when a heterozygous phenotype is distinct. So both the recessive and dominant alleles are contributing to that phenotype. So both of the parents' passed down alleles are expressed in the phenotype. We don't have sort of dominance where if they're heterozygous and the dominant alleles winning out to determine the phenotype. Epistasis is when many genomic loci and their alleles interact with each other, but all are contributing to the same trait. We've talked about eye color or hair color, so many different loci are adding together to determine that characteristic, that phenotype in the individual. And genomic loci can either be synergistic or antagonistic, so they can work together in the same direction and be additive, or they can be antagonists to each other and like mitigate each other out, or like reduce the effects of each other. And then pleiotropy is the opposite of epistasis, where a single locus is responsible for many traits. This locus is often called a master regulator. And a good example is to remember the time we were talking about measuring our feet with like our arms, which was kind of silly. But essentially it's just saying like, the same loci are controlling the size of different parts in your body. So you can imagine if you get some mutations of that genomic locus, that you can have some very detrimental effects on an individual. And these loci tend to be conserved because as you can imagine, for a locus to be responsible for many traits, like it's obviously very important in an organism in general, especially when we talked about like hip size and head size being connected. Norms of reaction are graphs looking at how a phenotype is expressed in different environments. So you're taking an individual, you're not altering their genetic like sequence or the way that the genetic sequence is being expressed yourself. You're just putting them in a different environment to see how the phenotype changes. So we have plasticity or being canalized. So plasticity is when the same genome leads to different phenotypes in a different environment. Dogs are a good example. So, they're all dogs, but they can look very different depending on their environments and what they're bred for, things like that. So their genome responds very rapidly to these sorts of changes, but they still are the same species. And then canalized is essentially, I guess, in contrast to dogs, could be cats. So I mean, they all pretty much look the same. They're all cats, so the same genome leads to the same phenotype in any environment. Speaking of the environment, so epigenetics is the influence of the environment on gene expression. Here we talk about methyl and acetyl group modifications. So when we talk about epigenetics, we're talking about the environment essentially causing chemical reactions in our bodies to change based on those environmental factors, which in turn can cause a gene to be on or off. So a gene being on means that the chromatin called euchromatin is open. You have unmethylated cytosines and acetylated histones. So this can go through the central dogma and transcription to produce a protein. A gene is off, that's heterochromatin. It has methylated cytosines and deacetylated histones. It's transcriptionally silent, so the sequence isn't altered in any way. It's just not producing that protein. The DNA here is so wound up it cannot be accessed to be transcribed, so you're not producing that protein. And the good example is to remember the Himalayan cats or the bunnies, that gene. So In Himalayan cat, for example, you have a very light colored core body, because there the protein for that darker color is not being produced, because it's a warmer temperature than the extremities like the ears and the paws. So at the ears and the paws, the temperature is colder. So it's unmethylated. And so the chromatin is open and able to be transcribed in order to produce that pigment protein. Next, you need to be familiar with making a 2-gene cross. And definitely some formulas that you will need to know for this exam, such as 2n+1, where n is the number of diallelic additive loci, and then using 2n+1, you will have the number of phenotypic classes. So an example here is 16 loci for human eye color, for example. That would be 33 phenotypic classes for eye color. So it can really tell you the complexity of all of these different loci, and how they can create a spectrum of different phenotypic presentations. Next is all about genetic linkage. Genetic linkage is, we're talking about how likely are alleles going to be crossed over. So alleles that are close together on a chromosome have a lower probability of crossing over during meiosis, so they may be linked. So the max amount of recombination frequency is 50%. And if it's way lower than 50%, you have genetic linkage. And if it's higher, then that suggests that those genes are unlinked, which supports Mendel's law of independent assortment, where genomic loci sort independently in meiosis into different gametes. And using genetic maps, we can describe allele order and distance, and we measure these genetic distances in centimorgans. 1 centimorgan is one like recombination frequency. And on the right, I have given you an example. So here I have one individual going through meiosis 1. So remember, we will have crossing over in prophase 1. So it's important to draw the parental and the recombinant gametes, which I've done here. So the parental gametes, you can see nothing about the allele has changed. They had originally a big F and a little g together on the same chromosome, or on the same chromatid if you would like. On the other one, they have a little f and a big G, so they would be heterozygous for both of those traits. And then of course we will have two two chromatids crossing over, and then we would have recombinant chromosomes. So here I have a big F and a big G and a little f and a little g, and notice that was not present originally in this individual, so crossing over occurred there to produce those. So now I'm going to take this individual's gametes, and I'm going to perform a test cross with a homozygous recessive individual for the loci of interest. And because I know the test cross is homozygous recessive, I know that they're only producing one kind of gamete, in which there's both of the alleles here, for example, are lowercase or recessive, but just know I'm crossing them 4 times. And then we can look at the offspring of this individual, of these individuals, for instance. So you will notice I just kind of made up some numbers. So there's more parental offspring, uh, offspring that have had passed down the original parental gametes, especially from that first individual with the parental recombinants. So there's more of those than of offspring who have a recombinant allele. So from this, we can find the map distance. And so map distance is the number of recombinants divided by the total number of offspring. So I've done 12 plus 9. And then I've added up all of the offspring, that's 110, multiplied by 100%, and you get 19% recombination. This is 19 centimorgans. That's the distance between these genes. So the genes are likely to be genetically linked because they're quite lower than 50%. The smaller the percentage, the tighter the linkage. So these genes are likely to travel together. So recombination, crossing over is less likely to happen. Next is heritability, where you definitely need to know some equations to calculate heritability. But luckily, if you kind of go through each individual equation and try to reason through why it's set up the way it is, you might find that helpful to remember. So heritability is how much variation of the phenotypes of a population is caused by the variation in genetics and the variation in the environment, which is definitely a mouthful. But essentially, you're looking at the variation of the phenotypes in a certain population, and you're asking yourself, well, certainly there's a lot of variation between everyone's genome there. And also, they could have incredibly different environments that they live and work in. So how much variation of both their genetics and the environment that they live in contributes to the variation of their phenotypes. So if all phenotypic variation in a group is caused by genetic factors, then heritability is 1%, is not 1%, definitely not percent, is 1. And if all phenotypic variation in a group is caused by environmental factors, then heritability is 0. So remember, when we calculate phenotypic variation, it should be a number between 0 and 1. So being closer to 1 means that it's being caused more by the genetic factors than the environmental factors. And if it's closer to 0, then environmental factors and their variation are playing more of a role. So phenotypic variance, you'll either see this as V sub P or V sub T. It's the total variance of the population. Genetic variance, V sub G, is variance due to variation among individuals' alleles. And environmental variance, V sub E, is environmentally caused variation. So phenotypic variance is just genetic variance plus environmental variance. And then another equation is for genetic variance. Genetic variance is the additive genetic variance plus the dominance genetic variation. So what is additive genetic variance? This is a variation that is due to the additive effects of alleles. And essentially the most important thing here is that note that this actually can be passed down from parent to child. Whereas in dominance genetic variation here is due to dominance relationships among alleles. So for example, if somebody is heterozygous for a certain gene, then that dominant allele is going to win out generally over the recessive allele and produce that certain phenotype associated with being dominant. But here, this cannot be passed down. This is restricted to your own genome because everybody can, have a different genotype, and like you're always going to get different combinations of whether or not there is even a a dominant allele to win out over the recessive allele, for example. But additive effects of alleles, this sort of variation can be passed down. So substituting the fact that variation, genetic variation is the sum of additive genetic variance and dominance genetic variance, then we can just kind of substitute these in. So total like phenotypic variation is additive genetic variance plus dominance genetic variance plus environmental variance. And for an inbred population, just use V sub T equals V sub E. It's just saying because there's no genetic variation, all of the variation, the phenotypic variation in that population has to be caused by the variation of the environment. Next, we have broad sense and narrow sense heritability. So broad sense heritability is the genetic variance over the total, like the phenotypic variance. Narrow sense is the additive genetic variance over the phenotypic variance. So why are these differences important? So broad sense takes into account both the additive and dominance variation, whereas narrow sense only looks into the additive variance that can actually be passed down. This is important because when we talk about heritability, I mean, we do want to know like how much of the variation in phenotype is caused by variation in genetics. So it's important to look at what can be passed down, especially if you're a breeder. So broad sense adds in that extra dominance genetic variation, which affects that number. It can skew it a little bit, which is generally why narrow sense heritability is more important in being more specific about the heritability. And it's more precise, but these numbers generally should be very similar, in kind of close to each other, towards the same end of the 0 to 1 spectrum. So narrow sense heritability again is just the additive genetic variance over the total, the phenotypic variance. And then next, another really important thing with narrow sense heritability is a response to selection. So here, if you want to measure a specific trait of a population and, for example, like how likely is it to be passed down? Maybe you're like a breeder or something. Let's go like with height as an example. So what you'll do is you'll take a population, you'll measure their height, you'll select a group from that population, measure their height, and then you will have that selected population breed. And so you want to compare the height of the next generation to the height of the selected group. So you just do the next generation's height minus the original like large population height over the selected population height minus the original big population's height. So this compares the next generation to the original to understand if the trait is worthwhile for breeding. Because you really want to know, like, is this sort of likely to stick around, or is there something else? Like, is it just not likely to be passed down? Maybe it's something that's quite affected by the variation in the environment rather than variation in genetics. And in that case, it's a lot harder to deal with because environments can be very, very hard to control. Whereas, I mean, our gene sequences generally stay the same for the most part, and there's a much greater amount of control when you are looking to breed. Lastly, this is what the article talked about. So missing heritability. So not all quantitative trait loci have a major impact on the phenotype. S- So because we have a lot of different QTLs that affect, like the same sort of trait, there can be, a big spectrum of phenotypes, 2n+1. So to see all of the genetic variation for a certain trait, you'd have to essentially sequence everybody's genome to account for, even the rarest phenotypes. So scientists always essentially underestimate narrow sense heritability. Um, it is truly difficult to get a good grasp of just how much genetic variation can play in variation in phenotypes across the population. Um, so if we can use a better model organism like yeast, for example, that can eliminate missing heritability because we can have more of a control on that, and so it's easier to look at scientifically. So that was the end. Let me know if you have any questions.